Okay, well, thank you very much indeed. It's a very great honor to be invited to give this sixth lecture in the M.S.Swaminathan Centennial year series. I knew Dr. Swaminathan in several different capacities. When he was the Director General at IPGRI, I met him several times at the time when I was the Deputy Director General of ICADA. And we worked particularly closely together in areas related to policy, genetic resources policy, ethics and equity issues related to genetic resources, and so on. He was a great scientist and a great human being. And I remember him coming to visit us one time and having dinner with us and meeting our young daughter, who was 8 years old at the time, who remembers him very fondly and often mentions him. Yeah, I've called this talk "Seeds on Ice," and you'll see why, particularly when we come to the end of the lecture. But it's really about why we need to conserve it and what steps are being taken to conserve. Just give an overview of the talk. What I'll talk about a little bit is about how growing populations, changing world situations, as increasing dramatically our need for new crop varieties. And I'll talk a little bit about how the role they can play. And I think this is probably going over some of the earlier lectures in this series, but perhaps just to recap. And then ending up with really a number of initiatives, both in the technical area, the policy area, the financial area, initiatives which have taken place mostly within the last 20 years, many of which are ongoing, but which I think give us hope that we can indeed feed the world of the future. Yeah, I'll start with just a few facts and figures, which I'm sure most of the audience here are familiar with, but just to set the scene, just to see what it is that we are facing, what are the challenges in trying to feed the world of the future. In 2023, it was estimated that approximately 2 billion people in the world were either moderately or severely food insecure. That means that they couldn't rely on necessarily being able to have their next meal. 1 in 11 faced hunger, and that's 1 in 5 in Africa. In 2022, it was estimated that 69 million children under the age of 5 were stunted. Now, a stunted child means that that person is set back for their entire life. In 2022, 5.8% of adults worldwide were considered to be obese, the opposite end of the spectrum, and reflecting severe issues related to food quality and quantity and distribution. The world population is expected to peak at about 10.3 billion people in the 2080s. That is about 2 billion more people that will have to be fed over the next 50 years than are alive in the world today. A massive task. And it's estimated that something like 50% will be an increase in global food demand of 57% compared with 2020 by 2050. But we also know, and this is a commonly cited figure, a third of all food produced is wasted. And of course, our natural resources are under extreme pressure. Per capita fresh water is diminishing, and agriculture currently uses approximately 75% of the world's fresh water. It's also been estimated that some 33% of global agricultural soils are degraded. Right, the next slide, please. And of course, given all of these rather depressing facts, we have on top of all that probably the biggest elephant in the room, and that's climate change. As probably many of you know, there are a number of winners and losers. Clearly, some of the winners might be Northern Europe, North America, East Asia. But we're seeing already significant impact, significant losses of agricultural productivity in the Amazon, Southern Africa, around the Mediterranean, particularly the North Africa region, in South and Southeast Asia. And overall, the losers significantly outweigh the winners. I'd just like to draw your attention to the map at the top right of the screen. And as you can see, there's a series of dots there. Each of those dots represents a point on the globe where a meta-study of 30 diverse climate models based on very different premises, 90% of them agreed on that figure, on that percentage change that's likely to be affected in annual precipitation. So I think we can see that the science is beginning to come together. I think we're beginning to see where the winners will be, and particularly the agreement on where many of the losers are likely to be. So this presentation will discuss the key role of the need to conserve and use crop biodiversity and how it's being done to help ensure our children and our grandchildren can eat a healthy diet. First of all, why is diversity important? And of course, diversity exists on many different levels. A diverse diet is considered an extremely important part of health. Human health depends very often on a diverse diet, of having access to a wide range of different nutrients. Diversity, of course, can confer resilience in farming systems. In general, the more diverse a system is, the more resilient it is. And of course, a lot of the diversity of particularly the older heritage crops that we have a very big historical and a heritage and a cultural value that I think shouldn't be underestimated. But next slide, please. Perhaps the most important use for diversity going forward is in its underpinning of crop improvement, of creating those varieties that we will need to face these new conditions, new higher temperatures, perhaps waterlogging, cooler temperatures in some areas, drought. So varieties which have a higher and more stable yield, stability being an extremely important factor. With greater pest and disease resistance, we're going to see a shift. We're already seeing a shift in the spectrum of pests and diseases around the world. Varieties with improved nutritional and processing qualities, varieties that will lead to novel foods, and particularly varieties that are adapted to higher temperature, to drought, waterlogging, salinity, and many other environmental negatives. We're looking for varieties that will have increased resource use efficiency, that are able to use nitrogen, able to use water more efficiently. We also need varieties which result in reduced greenhouse gas emissions. Particularly, we're looking at gas emissions from paddy fields and from livestock, and it's possible already. There's some interesting work being done on forages that result in less methane emissions from ruminants. We also need crops that will have perhaps deeper rooting systems and able to have greater carbon sequestration. And of course, with climate change, not only do we see long-term climate effects, but an increase in extreme weather events, and resilience in the face of such extreme events is becoming increasingly important. So when we're talking about breeding, where does this genetic diversity come from? With a variety of sources, from land races, farmers, domesticated varieties, with domestication having gone on for some 10,000 years. And it's a process which importantly still goes and continues today. Farmers around the world are continuing to select and adapt material to meet their ever-changing conditions. Crop wild relatives with modern genetic technologies, the progenitors of today's crops and their close wild relatives are becoming increasingly important as a source of genetic diversity. Commercial varieties, currently grown and also obsolete varieties, also contribute very important traits, genes to the plant breeding pool that breeders can work with. And of course, increasingly genetic stocks induce mutations, breeding lines, mapping populations, and so on are becoming extremely important within the whole context of genetic improvement. But of course, it's widely known we're losing crop diversity at a very large rate. Diversity is threatened at many different levels. It's threatened in the wild, of course, as habitats are degraded or lost due to climate change, due to increased urbanization, due to infrastructure construction, airports, roads, etc., and of course, deforestation and land degradation. In a recent study, a meta-study looking at 33 articles from 1988 to 2020, 90% of these articles reported a loss of genetic diversity of crop wild relatives in their natural habitat. Likewise, on farmers' fields, as farmers change their systems to meet new demands, switch to new varieties and new crops that are more economic, and as consumer demand for different foods change. And again, this same article looked at 139 articles from 1930 to 2021 and found 86% of these articles reported a decline in the diversity of land races on farm. And of course, we're losing it even in gene banks. Gene banks that were set up to safeguard this genetic diversity, in many cases, in many parts of the world, are unable to fulfill this due to natural disasters, war, civil strife, and particularly just through lack of funding and mismanagement. Many countries of the world, their national gene banks are extremely strapped for cash and resources and are able to be able to guarantee to safeguard the material within them. In fact, again, this same article looked at 28 papers from 1995 to 2021. 85% of these documented the loss of genetic diversity actually in gene banks. And it's not only a national issue, it's a global issue. Why is genetic diversity a global issue? Well, no country is fully independent with respect to the genetic resources it needs to improve its agriculture. There's a very high level of interdependence among nations for genetic diversity. It's estimated that approximately 70% of national food supplies originate from crops that were domesticated elsewhere, from crops that originated elsewhere. And likewise, almost 70% of crops that are grown naturally on farms originated in other parts of the world. That's one reason why interdependence among nations is so important. The other is that when we look at a modern variety, and we look at where the parental lines have come from, we find that particularly for crops with a long breeding history, such as wheat, we find a massive number of different inputs going into a modern wheat variety. I put here a pedigree of Sonora 64, which was released in 1964, and even as long ago as that, there are approximately 30 land races from 15 countries in the pedigree of this early Green Revolution wheat variety. Even in the 1980s, the Vary wheat lines, which are a very important series of crosses that led to many, many varieties around the world by CIAT, includes more than 3,000 crosses between 51 parents from 26 different countries in 5 continents. A huge, massive input from so many different parts of the world into a single important variety. So, given this loss, given the importance of this material, what can we actually do about it? Well, there's several different approaches, of course, we can take. In situ conservation, the conservation of materials in wild areas and protect under special management regimes, perhaps around farm margins of fields on farms and the like. Particularly important when we're trying to conserve the wild relatives of crops, the progenitors of our crops, which can contribute so much to new varieties. We can encourage and support farmers to maintain traditional varieties on their farms. And how can we do this? Well, either through direct payments, which is tried, but perhaps not a particularly reliable long-term solution. The longer-term solution would be to make sure that these traditional varieties are economic for farmers by expanding markets, by increasing demand for perhaps novel foods and the like. And what I'm going to concentrate most of the rest of this talk on is ex situ conservation in public and private gene banks at different levels, at the local and community level, at the national level and regional international level. And perhaps just a very brief mention of increasingly, it's not just the physical material in gene banks that's important, but the so-called digital gene banks. Banks in silico banks maintained on computers. It's the genetic, the genomic, phenomic information and so on that is becoming increasingly important. And of course, if we're looking at conserving a gene pool of any crops of interest, we will need all approaches. We need to integrate in complementary approaches if we're going to be able to conserve the maximum amount of diversity within the gene pools of interest. So why ex situ conservation? Why is it important? Unlike in situ conservation and on-farm conservation, ex situ conservation halts natural selection. So ex situ conservation on-farm is extremely important. It allows material not only to be conserved, but also to evolve to meet changing conditions. Ex situ conservation, when you put material in a gene bank, its evolution essentially ceases at that point. Why is it important? Well, of course, even in the wild and on farm, material is still being lost. And already many materials that you'll find in gene banks no longer exist in the wild or can only be found in gene banks. It provides a safety net. If there's a natural disaster, a man-made disaster in the field or in other gene banks, then the ex situ conservation allows for reintroduction. But I would argue probably one of the most important, one of those valuable aspects of gene banks and ex situ conservation is that it provides a readily accessible source of genes, traits, genetic information that underpins crop improvement. It means that we don't need to continually recollect from the wild every time we need a new parental line. And gradually one can build up huge amounts of data and information on these collections that would not be possible without them. Yeah, so ex situ conservation can occur in many different ways. In seed banks, field gene banks, where plants are grown out in the field. It could be going in vitro in test tubes or in petri dishes in laboratories. Cryopreserved in liquid nitrogen, and increasingly gene banks are looking at other ways of storing things like pollen, lyophilized leaf samples, and so on, freeze-dried leaves, etc. Next slide, please. So a little bit about gene banks around the world. Today, there are approximately 1,700 gene banks worldwide, and of increasing importance because of the growing importance of crop wild relatives and other non-cultivated species. Botanic gardens are becoming increasingly important as a source of genes and traits for crop improvement. Of the 3,000 botanic gardens worldwide, approximately 350 in 75 countries have got seed banks, a very important source that's often overlooked by the agricultural community. It's been estimated that approximately 5.8 million samples are held under medium-term and long-term storage. That's at plus 5 degrees, considered medium-term storage, and longer-term at minus 18 in deep freezes. Of this material, the origin and its biological status is known for only about 70% of these. So a large percentage of material that we have in our gene banks, we really don't know that much about in terms of its origin and biological status. However, we are able to build up a growing body of data about its characteristics, its evaluative data, and the like. Yeah. Of the 70% of accessions where we know their biological status, 35% of these are farmers' varieties, traditional varieties and land races, about 18% crop wild relatives and other wild species, about 1% of wild food plants, that is plants that are actually harvested directly from the wild for their food value, and the remaining 46% are mostly improved varieties and breeding materials. So we see quite a spread there. But if we look to the future, we probably need to shift that focus a little bit more in favor of the traditional varieties and crop wild relatives. And I'll talk a little bit later about how we see the future of ex situ conservation. And if we look at the mix, the makeup of the crops within those 70% of the 5.8 million accessions, we see that the large majority are cereals, followed by pulses and forages. And then we get down to relatively small numbers when it comes to vegetables, fruits and nuts, oil plants, herbs, spices, roots and tubers and others. And particularly to note here, the roots and tubers, only 2% of the conserved material were roots and tubers. And I'll come back to that in just a moment. Just an example to give some idea of the numbers of that we're talking about here. And I've used here just some of the collections that are maintained by the CGIR centers, data from 2021 that would not be significantly different today. That's the barley collection of IPGRI, maintained now in Morocco, and it's approximately 32,000 different samples of barley. CIAT, the International Centro of Tropical Agriculture in Colombia, has a collection of 38,000 different types of beans. Chickpea between IPGRI and ICRISAT in India, some 34,000 accessions and so on. I won't go down that whole list. You can see for yourself, but the two largest collections are rice, and particularly maintained by Africa Rice at IPGRI, and actually the CIAT should be included there too, is almost 150,000 different samples of rice. And wheat, with the collections between CIMIT in Mexico and IPGRI in West Asia and North Africa, have almost 200,000 different types of wheat, in different samples of wheat in that collection. The CGR distributes approximately 100,000 germplasm samples per year to something in the order of 120 different countries. So a massive effort that underpins, is one of the major underpinning of crop improvement worldwide. Yeah, so I mentioned earlier about roots and tubers representing only 2% of the total global accessions held ex situ. And the reason for that is that 20% of all crops cannot be stored as seed. And that's either because they're clonally propagated, such as banana, cassava, potato, sweet potato, yam, some extremely important crops that cannot be conserved as seed, even though it's possible to conserve seed. The genetic integrity depends on clonal propagation. And the second group are recalcitrant seeded crops. And these are crops in which the seed cannot be desiccated and frozen. They die. So they cannot be held in conventional cold storage stores at gene banks, such as can, for example, with cereals and most food legumes. And again, some extremely important crops in this category: breadfruit, cacao, coconut, coffee, jackfruit, taro, tea, and many of the tropical fruits, and even some of the temperate fruits, cannot be stored as seed. And as pointed out, approximately 90 accessions worldwide are maintained as seed, with most of the remaining 10% in either field collections or in vitro collections, grown as plants maintained in the field or maintained as tissue cultures in test tubes or Petri dishes. Both techniques are expensive and both techniques are insecure. Plants growing in the field or in vitro are liable to be attacked by pests and diseases. In case of field collections, inadequate provisions for irrigation and so on can also result in loss. The only really safe way to conserve these is through cryopreservation. That is conservation in liquid nitrogen at 196 degrees centigrade. Cryopreservation is the equivalent of the long-term storage for seeds, but it's still considerable more research is needed. Not all crops can be cryopreserved. We don't have robust protocols for many of the crops that can't be stored as seed, and facilities are expensive and need to be developed. Recently, a global plant cryopreservation initiative was started by the Crop Trust, various CGI centers and others to try and develop a system of a network of cryopreservation facilities that would offer a kind of backup for collections of clonal crops, recalcitrant seed crops, in a way analogous to the Svalbard Global Seed Vault, which I'll talk about in a few minutes. So what does the future of gene banks look like? With our rapidly growing ability to use genetic resources and genomic information through the use of gene editing, synthetic biology, and the like, through our growing ability to generate evaluation data on many, many different new traits through high throughput phenotyping and many other new biochemical and techniques. Our ability to generate genomic information, next generation sequencing, the like, which is getting ever more accurate and cheaper. And of course, our increased information handling systems, and particularly the growing use of artificial intelligence, means that I think the gene banks in the future will need to concentrate much more of their attention on the collections of crop wild relatives, of crops that today are perhaps not very important, often referred to as underutilized crops, increasingly referred to as opportunity crops, crops that may come into their own with changing climate and demand patterns. And of course, as I've just mentioned, increasing concentration on clonal recalcitrant seeded crops, which are so underrepresented in gene banks today. Special trait collections, I think, are going to become more important. Collections of plants which exhibit particular systems of drought or tolerance or heat tolerance and so on. So as our ability to gene edit and understand biochemical processes, I think will lead to an increasing value of special trait collections. Of course, special collections of special genetic interest. Having done a lot of this work, material and information, genetic and phenomic information known about particular exceptions makes them ever more valuable and that they and worth conserving, including various mutants, mutant stocks, mapping populations, genetic stocks, cytological stocks, advanced breeding lines, and the like. And of course, increasingly, gene banks are going to have to be developing their information links. And in order to ensure this material is not only conserved, but much more widely used, the information systems need to link and be made useful for farmers and for plant breeders, for scientists, and not us just sharing information with other gene banks. Okay, just a bit of a diversion away from the sort of the technical to look a little bit about the issues of owner and access and benefit sharing. We've talked a lot about this genetic diversity created by farmers, diversity created by breeders, natural diversity. Who owns this diversity? Who has the right to access this diversity, and on what basis? And these are issues that have been, I think, at the forefront of the genetic resources of this world for, I think, at least 40 years. The Keystone Dialogue series on plant genetic resources that took place between 1988 and 1991 was chaired by M.S.Swaminathan himself, was an extremely important series of dialogues that involved the public sector, private sector, NGOs, farmer groups, researchers, national, international, gene bank managers, and the like, trying to thrash out the issues of who has ownership of this material, who has access to this material, and what happens if someone wants to patent material, for example. Should that be allowed? Should that not be allowed? And if so, under what circumstances? These were dialogues that were taking place at the time when negotiations were underway that led to the Convention on Biological Diversity. The Convention on Biological was adopted in 1993 and was, I think, in many ways a landmark for understanding the ownership issues and the basis on which access would be provided through negotiation, and with adequate benefit sharing arrangements, where the benefits of using this material flow back to the countries and the communities that originally led to that originally developed that particular diversity. And although the Convention on Biodiversity, I think, was a landmark particularly for bioprospecting in the pharmaceutical industry, which had led to a lot of the discussions, it wasn't the ideal solution for the agricultural world. And as the Keystone Dialogue series on plant genetic resources had pointed out, the agriculture has a very different need from pharmaceuticals. And as I pointed out earlier, when I showed that pedigree, for example, strictly bilateral negotiations and discussions were not appropriate in many cases for agricultural biodiversity. There was a lot of concern at the time that particularly the international collections maintained by the CGIAR were being treated by the CGIAR as if they were their own material, and they had their own rights to use them in any way they wished. This was creating a lot of concern among various countries and NGOs, and particularly those countries from which a lot of this material had originated. And this led in 1994 to agreements being signed between the centers and FAO, in which the centers declared that they were not owners of these collections, but they were trustees of these collections, that they held these important collections in trust for the global community. As a result of these agreements, the CGIAR set up the Genetic Resources Policy Committee, which operated between 1994 and the early 2000s, and until the year 2000 was chaired again by M.S.Swaminathan. The idea of this committee was to oversee the in-trust agreements. It coordinated the input of the CGIAR into the development of the international treaty that I shall talk about in a moment, and looked at issues of ethics and equity, particularly holding a workshop in Brazil in 1997, which led to the statement on the policy on ethics and equity adopted by the CGIAR in the early 2000s. Can I have the next slide, please? But all of these concerns led to a realization that a special treaty was needed to cover the genetic resources of agricultural crops. And as a result, the International Treaty on Plant Genetic Resources for Food and Agriculture, which was negotiated over a period of about, I think it must have been about 11 years, finally came into force in 2004. It's fully consistent with the Convention on Biological Diversity, but tailored to the needs of agriculture. In that, whereas the Convention on Biological Diversity tended to promote bilateral agreements between countries and users, the approach taken through the international treaty was one of a multilateral approach to access and benefit sharing. And as of January of this year, 152 states plus the EU have ratified the treaty, which covers all aspects of conservation and use of plant genetic resources for food and agriculture, and makes a particular feature of farmers' rights, the rights of farmers that arise from their having developed so much of the material that is used in agriculture around the world today, and to make sure that they, particularly farmers in developing countries of the global south, have direct benefit from their own efforts at conserving and using genetic diversity. Although the treaty covers all plant agricultural crop diversity, it creates a specific multilateral system that covers 35 crops and 30 forage species. And for these particular species, it develops what is referred to as the multilateral system of access and benefit sharing. It also covers the international collections held by the CGIAR centers and other international centers that have signed up to the treaty. The access regime means that all signatory countries are able to access material that's covered by this multilateral system, and with a mandatory benefit sharing provision that if any of them take out any sort of intellectual property that restricts the subsequent use of this material, they're required to pay 0.7% of sales into what is referred to as the benefit sharing fund. The benefit sharing fund, which has a fund that is funded both from the mandatory payments, but also, much more importantly, voluntary payments, is used then to support the conservation and use of genetic diversity in developing countries. And although I think the international treaty was in many ways a breakthrough, and particularly for agriculture, it allowed materials to be exchanged and moved much more freely than had been possible under the Convention on Biological Diversity regime, it's still got a way to go. And negotiations are still underway, looking at ways in which the coverage of the treaty can be expanded to cover all PGRFA, not just the 35 crops and 30 forages. Particular concerns about digital sequence information. Who has the right to use this? How will, and what benefits should be paid on using not necessarily the actual physical material, but of course the information on that material becomes extremely valuable in its own right. Farmers' rights and benefit sharing still need to be further expanded and built upon. Yeah, and it's not only policy aspects that are important, but also funding. The concerns were growing, particularly during the 1990s, that these important collections were being inadequately resourced, both at the national level and at the international level. The centers were beginning to suffer quite severe funding shortages and weren't sure how they were going to be able to maintain these collections, even though they had legally agreed to do so in the agreement signed with FAO. I remember one time I was at IPGRI as Director General of IPGRI, and I received a call from a national gene bank that for the sake of $5,000 was going to have to switch off the electricity, couldn't afford to pay the electricity bill and risk losing its entire collections as a result. Well, with these sort of issues in mind, the Crop Trust was founded in 2004, essentially to fund and help support the global system of ex situ conservation in perpetuity. Now, part of the problem facing national and international gene banks was increasingly donors were segmenting their support into sort of 3-year project cycles, so that funding for longer term, planning for the longer term, such as needed by gene banks, was becoming increasingly difficult. And the solution to this was seen to be an endowment fund from which the offtake would be guaranteed while the capital maintained. As a result of this, the Global Crop Diversity Trust was established, the Crop Trust, which now to date has an endowment of approximately 350 million, which allows for a $9 million annual offtake to support gene banks around the world, but has a target of 600 million. At 600 million, the offtake would be sufficient to support all Article 15 gene banks, all gene banks, international gene banks around the world. And a further target beyond that would be looking to support national collections, and particularly national collections of international importance. Next slide, please. Yeah, to date, the Crop Trust has provided long-term grants for 18 different crops in 11 different institutions, but still obviously has a way to go. It also funds special projects looking at issues such as regeneration, wild relatives, opportunity crops, and the like. It's supporting important information systems like the Genetic Resource Information Network Global, which is the community edition, which was recently released, is particularly valuable for managing gene banks, and Genesis, an international system for maintaining information on characterization and evaluation data of the world's germplasm. It also supports the last topic I'm going to cover briefly, and that is the Svalbard Global Seed Vault. Next slide, please. What is the Svalbard Global Seed Vault? Well, it's a unique facility that's been developed as a backup for the world's gene banks. It's not a primary gene bank, but it exists as a gene bank where other gene banks can deposit their material in a safe location and will remain available for reintroduction should it ever be needed. It's a backup safety net developed by the Norwegian government with the Norgen, the Norwegian Genetic Resources Institute, and funding from the Global Crop Diversity Trust. It opened in 2008 on Spitsbergen, which is the Svalbard archipelago. You can see where it is well north of the northern end of northern tip of Norway, 78 degrees north, 1,300 kilometers from the pole, and located about 130 meters above sea level. So with sea level rise, it still would remain safe. So this just shows it's located in the permafrost, but is artificially cooled to minus 18 degrees, the standard temperature for long-term conservation. It's in 3 vaults set in 100 meters into the side of the mountain. Currently has a capacity of 4.5 million samples based on 500 seeds per sample. This whole facility cost around $11.3 million to build. The original building for about $8 million, and subsequently some upgrades for a further $3.3 million. And its annual running cost is about $500,000. So about 30% of the world's conserved plant genetic resources are safety duplicated, which is very low, and that figure needs to be much higher. But of that 65% are in Svalbard, at least as one of the locations where they're duplicated. As of February this year, the seed vault housed 1.33 million different seed samples of 6,297 different species. So a lot of those are crop wild relatives as well as crops from 123 different gene banks around the world, located in 91 different countries. And it's already proved its worth. The Carter collection was put in the seed vaults in, I think, around 2010, 2012. With the civil strife in Syria, the collections there were lost, but the material that was stored in Svalbard was able to be restored and has grown out in Morocco and Lebanon, and those collections have been able to be regenerated and survive as a result of the global seed vault. I would argue that this is probably arguably the world's best insurance policy. At $500,000 premium a year, it's hard to imagine anything that could be better. Next slide, please. So, in conclusion, genetic diversity underpins food security. It enables, it provides resilience in today's agriculture, but it's particularly important in enabling agriculture to adapt to the many future challenges that it will face. Much has already been conserved, but as I've pointed out, much remains under threat, not only in the field, on farmers' fields in the wild, but also on the gene banks that are underfunded. I mentioned a number of technical, scientific, financial, and policy developments that have occurred mostly over the last 20 years or so. And I think many of these give us hope, but we still have a long way to go. And unless we're successful in this task, we will severely limit our options for meeting global food security and will impoverish future generations in so many different ways. Thank you very much for your attention.